Hello everyone, welcome to Chapter 3. In this chapter, we are going to learn about prokaryotic cells and microorganisms. As we learned in Chapter 1, the cells of life are classified into prokaryotic cells and eukaryotic cells. Eukaryotic cells are found in animals, plants, fungi, protozoa, and algae. Prokaryotic cells are found only in the bacteria and archaea. Cyanobacteria, actinomycetes, mycoplasma, chlamydia, rickettsia belong to generalized bacteria. When compared to eukaryotic cells, the structure of prokaryotic cells is less complex. The chief characteristics of prokaryotic cells are as follows. First, typically their DNA is not enclosed within a membrane and is usually a circularly arranged chromosome. Second, their DNA is not associated with histones. Third, they generally lack organelles. Advances in microscopy reveal a few membrane-enclosed organelles. However, prokaryotes lack other membrane-enclosed organelles, such as nuclei, mitochondria, and chloroplast. Fourth, their cell walls almost always contain the complex polysaccharide peptidoglycan. Fifth, they usually divide by binary fission, where DNA is copied and the cell splits into two cells. In this lecture, we will learn to talk about bacterial shapes and arrangements, bacterial sizes, and structure of generalized bacterial cell. The common shapes of bacteria are spherical, rod-like, and spiral. If the cell is spherical or ball-shaped, the bacterium is described as a coccus. They can exist singly or can be associated in characteristic arrangement and can be useful in their identification. The main factors influencing the arrangement of a particular cell type are its pattern of division and how the cells remain attached afterward. The greatest variety in arrangement occurs in cocci. They may exist as singles, in pairs, in tangents, in irregular clusters, or in chains of a few to hundreds of cells. When cocci divide to reproduce, the cells can remain attached to one another. Cocci that remain in pairs after dividing are called diplococci. Those that divide and remain attached in chain-like patterns are called streptococci. Cocci that divide in two planes and remain in groups of four are known as tangents. Cocci that divide in three planes and remain attached in cube-like groups of eight are called cisternae. Cocci that divide in multiple planes and form grape-like clusters or broad seeds are called staphylococci. These group characteristics are frequently helpful in identifying certain cocci. A cell that is cylindrical is termed a rod or bacillus. Rods are also quite varied in their actual form. Depending on the bacterial species, they can be blocky, spindle-shaped, round-ended, long and thread-like, or even clubbed or drumstick-shaped. Bacilli divide only across their short axis, so there are fewer groupings of bacilli than cocci. Most bacilli appear as single rods, called single bacilli. Diplobacilli appear in pairs after division, and streptobacilli occur in chains. Some bacilli look like straws. Others have tapered ends, like cigars. Still others are oval and look so much like cocci that they are called coccobacilli. Spirillum are rigid spiral-shaped cells. Many have tufts of flagella and one or both ends. Bacteria that look like curved rods are called vibrilles. Typical examples of vibrilles are Della-vibrio and Vibrio cholerae. A bacterium having the shape of a spiral-shaped cylinder is called a spirillum. A rigid helix twists twice or more along its axis. Typical example of spirillum is Campylobacter jejuni. Spirochetes are flexible, spiral-shaped bacteria that have a unique internal flagellar arrangement, such as the leptospiral interrogans. In addition to cocci, bacilli, and spirilla that we just learned about, some bacteria are filamentous, which takes the form of long filaments. The most common filamentous bacteria are actinomycetes. Some bacteria have special morphological characteristics, such as star-shaped and rectangular prokaryotes. After we learn about the shape of bacteria, let's move on to the size of bacteria. The cell size of prokaryotic cells varies greatly in different species. In some prokaryotes, the cell size is similar to the largest virions. The sizes of some prokaryotic cells are similar to algae cells and can be recognized almost by naked eyes. The size of bacteria is measured in microns. Generally, the diameter of cocci is 0.5 to 1 micron. The rod-shaped bacteria is 0.5 to 1 micron wide by 1 to 3 microns long. Escherichia coli is an excellent representative of the average size of bacteria. As the figure shows, this rod-shaped bacteria is 1.1 to 1.5 micron wide by 2 to 6 microns long. However, the size range of bacterial cells extends far beyond this average. Near the small end of the size continuum are membranes of the genus Mycoplasma, and the other end of the continuum are bacteria such as some spirochetes, which can reach 500 microns in length, and the cyanobacteria oscillatoria, which is about 7 microns in diameter. The host bacteria lives in the intestine of the broad surgeoning phase. This bacteria grows as large as 600 by 80 microns, a little smaller than a printed hyphen, and clearly larger than the well-known eukaryote Promycea. An even larger bacterium, Sailmargarita namibensis, has been discovered in ocean sediments. Thus, a few bacteria are much larger than the average eukaryotic cell. This bacterium, usually 100 to 300 microns in diameter, occasionally reaches a size of 750 microns, 100 times the size of common bacteria. Sailmargarita namibensis use sulfide from bottom sediments as an energy source and nitrate. It appears hollow because it stores high concentration of nitrate in a central vacuole. We have learned about the morphologies and sizes of bacteria. Next, let's learn about the structure of generalized bacterial cell. Prokaryotic cells appear featureless and two-dimensional when viewed with an ordinary microscope, but this is only because of their small size. Higher magnification provides increased insight into their intricate and often complex structure. Bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. The innermost layer of the cell envelope is the plasma membrane, which surrounds the cytoplasm. Most bacteria have a chemically complex cell wall which covers the plasma membrane. Many bacteria are surrounded by a capsular or slime layer external to the cell wall. Because most bacteria don't contain internal membrane-bound organelles, their interior appears morphologically simple. The genetic material is localized in a discrete region called the nucleoid, and usually is not separated from surrounding cytoplasm by membranes. Ribosomes and larger masses called inclusions are scattered about the cytoplasm. Many bacteria use flagella for locomotion. Okay, that's all for today's lesson.